Hey guys, welcome back to Computational Biology with Python. In the last video, we worked through the Seq package of Biopython, and we got through the translate portion. So we were translating mRNA into protein sequences. So we're going to continue on with that line of work in this video. So let's jump on it. Okay, so at the end of our last video, we translated this RNA, and it had a premature stop codon, right? So the full sequence wouldn't be one long peptide chain because we had that premature stop codon. And so one thing you can do is you can specify the codon tables. So let me pull this up real quick. So if we look at a comparison here of a nuclear codon table versus a mitochondrial codon table, you can see, move over a little more, that there are certain codons, there's 4 or 5 of them, that are translated differently in mitochondrial genome versus the nuclear genome. So you can see AGG would normally be arginine in a nuclear genome, it's a stop codon in the mitochondrial genome, and UGA, which is normally a stop codon in the nuclear genome, instead codes for tryptophan, etc. So if we want to do translation of a mitochondrial genome, what we can do is coding DNA, translate, just like we did before, but we can say what codon table we want to use. And so we'll say vertebrate mitochondrial. And so now when you run it, you see that we get a different sequence that does not have this premature stop codon because it was a mitochondrial gene that we were actually sequencing. You can do the same thing with the codon tables by using the NCBI table number. So they have different tables published. If you find it, you can just do the codon or the table number, and it'll give you the same thing. So what if you want to translate the nucleotides up to the first in-frame stop codon and then stop and not? So for example here, we were like, okay, we translate to the stop codon, but then we kept going. But what if you just wanted to stop there? What we can do for that is coding underscore DNA dot translate, and then we say to stop equals true. If we do that, you can see it stops where the stop codon was previously up here. Should make this a little bit bigger. There we go. And then if we do, let's do it like this then. Coding DNA translate table equals 2, and then to stop equals true. What would you expect to happen here? Well, since the stop codon, there is no premature stop codon in the mitochondrial genome, you'd expect that we'd get the same thing, right? And we do. So you notice that it doesn't publish if you do to stop, it takes the asterisk for the stop away. All right. So if you want to, we can even modify this more. Say, let's do table 2, and say you want to specify what your stop codon is. You could make your stop symbol. Say we'll make our stop symbol an exclamation point. Oops, there we go. So we've replaced the asterisk with the exclamation point, which I don't know how much utility you'll find in that, but you can do it. So now suppose we have a complete coding sequence, CDS, which is to say a nucleotide sequence. But it's a nucleotide sequence after we've spliced out all the introns. And so this is a whole number of codons. So the length has to be a multiple of 3. So we can commence with a start codon and end with a stop codon and have no in-frame stop codons, right? So this is something that's been transcribed. Now, what if our sequence uses a non-standard start codon? Like I said, this can happen in a lot of bacteria, so it's not one of the 3, or one of the methionine that's normally in vertebrates. So how can we handle that? So we can do gene equals, and I'm just going to cut and paste this. Oh, I hope this pastes right because it's kind of funky. I might have to get rid of some of this stuff. All right, so we're gonna have this big long sequence here. There we go. Oh, let's get rid of this. Okay, so we have this big long nucleotide sequence here. And we have to make it a Seq object. Where'd my end thing go? Okay, let's see this. Hope this runs. Seq is not defined. Oh, got uppercase S. There we go. Okay. So we can do gene that translate table equals bacterial. And so we get our nucleotide sequence here. Oh, actually, hold on one second. Okay, that looks right. Now let's do gene translate table equals bacterial. To stop equals true. Oh, this has got to be like this here. Okay, so it published all the way to the stop codon here. So in the bacterial genetic code, this GTG is a valid start codon, and while it does normally encode valine, it is used as a start codon and should be translated as methionine. So this happens if you tell your bioconductor sequence it is a complete CDS. So if you don't tell it it's a complete CDS, it's going to translate it as valine, but if you tell it's a complete CDS, it'll know to do it as methionine. So let's do that. So gene translate table equals bacterial and CDS equals true. So that should tell Biopython that this is a complete gene, so it should not start with valine, it should always start with methionine, right? So it changes that to methionine for us, which is great. All right, so in addition to telling Biopython to translate an alternative start codon as methionine, it makes sure that your sequence really is a valid CDS. And so if it's not, then it will spit out an error for you. So where should we go from here? So let's look at codon tables here. So from bio.data import codon table. All right. So we can do standard table equals codon table dot unambiguous DNA by name, and then we'll say this is our standard. And then mito table equals codon table unambiguous DNA by name. So this is the function from the codon table package. Vertebrate mitochondrial. So let's run that one. Vertebrate. So, or you could just use the numbers 1 and 2 because they're numbered just like the NCBI ones. But this at least, sometimes it's nice to be able to use the 1 and 2s, right? Because it's less typing, and as you can see, I do typos a lot. But you want this also to be rereadable. So if you don't put in your annotations that the vertebrate mitochondrial is table 2, then when you go back to reread this in like 9 months, and I've had this problem before. So when I started coding, I didn't annotate very well. Then I'm like, oh, I should send the script to somebody, and so let me go reread it first. And I have no idea what I did, because it made sense at the time, and then you forget all the nuances. So, so be careful with that. Okay, so let's do, so we can visualize the tables by printing them, which is pretty cool. So print standard table, and it gives us our codon table, which is awesome. And then we can also do print mito table. We can see our mitochondrial table. And so I'm not a mitochondrial person in terms of doing research, so we can use these tables. We can say mito table stop codons and ask it what the stop codons are. And it'll say, okay, TAA, TAG, AGA, and AGG are the stop codons for the mito table. We can also do mito table start codons. We can ask, okay, what are the start codons? It says we have these 5 different start codons. So that can be useful, like especially, if as a reminder or as you're writing your program to look at mitochondrial gene expression, as a reminder or a print, or you could put it in there as, like I said, if you were to give the code to someone else, you can say, make an annotation that says we used the mitochondrial table because this is mitochondrial DNA and our start codons are these, and then just print them out. So let's talk a little bit about sequence comparison. So it's usually a very complicated topic. And there is no easy way to decide if 2 sequences are really equal. The basic problem is the meaning of the letters in a sequence are context dependent. So the letter A could be part of a DNA, RNA, or protein sequence. Biopython can track the molecule type. So comparing to Seq objects could mean we can do this as well. So should a DNA fragment have ACG and an RNA fragment have ACG, would they be equal? What about the peptide ACG or the Python string ACG? In everyday use, your sequence will generally all be the same type, so you're either going to be using all DNA or all RNA or all peptides. Sequence comparison only looks at the sequence and compares like in the Python string. So let's do Seq1 equals Seq ACGT seq1, no, sorry. So if we do ACGT, does that equal seq1? It says true. And it says seq1, does that equal ACGT? Yes, so those are fully equal, right? So using sequence objects as keys in a Python dictionary is equivalent to using the string as a plain string for the key. So the name of the seq is equivalent to the actual sequence inside the name, if that makes sense. Okay, so in some cases, you'll want to put in a code where the length of the sequence may not be known, what the actual letters are. Or so the length of the sequence is known, but not the actual letters, right? So a GenBank file may represent a genomic DNA sequence only by its config information without the sequence contents explicitly. So such sequences can be represented by creating a Seq object with the argument verb none followed by the sequence length. So for example, let's do unknown Seq equals Seq none. We know that it's 10 base pairs long. So we've created a well-defined length, and so any attempts to access the sequence contents, however, will raise this verb error. So if we do unknown Seq, it says we have a Seq with none in it. It's not really an error, but it'll give you this information. But it's length 10. And we can ask like length of unknown Seq, and it will say 10. Let's see. All right, I think that we're gonna stop there with unknown sequences. So, and you might say, like, why would I do that? Well, you could create this container for an unknown sequence of certain length and fill it later once you do your analysis, or say you're comparing gene sizes, and so you don't really need to know the actual base pairs, but you know the name of the gene and the size of the gene. You could store it as that variable, and I'm sure there are other applications. Those are just off the top of my head. So okay, we're getting through it. Yeah, there's so much stuff. So I'm trying to like parse it a little bit as we go, and figure out what might be interesting to you and what might not be interesting to you. But yeah, so that is it for Seq Objects. For this video, so I hope you watch it. I hope you watched it. I hope you liked it. If you're watching on YouTube, please hit like, please hit subscribe, and we'll finish out Seq Objects in the next video. So catch you in the next one.